Hi, this is Chetan, Facility Technologist in Sense IIC, and now we are in front of the Confocal Microscope tool, and this is the bio wing of Sense. So this Confocal Microscope is a higher end tool for the fluorescent samples. Basically, here the samples are tagged with the fluorochrome, then the image will be captured by this instrument. And whatever the instrument we have here is from Olympus, and its model is FE3000. This instrument has 4 excitation lasers, and which lies in the visible region. This microscope is a inverted and single photon microscope. This instrument is equipped with the two scanners, namely resonance scanner and galvano scanner. So galvano scanner will slowly scan the sample, and resonance scanner will scan very fast. So that resonance scanner will be used for live cells. So these are the contents I would like to cover with you. So basically, the fluorescence is actually a type of photoluminescence. So photoluminescence means when a light of certain wavelength is incident on a sample which is tagged with the fluorescent dye, so that sample will absorb the light and it will excite to the higher electronic energy states, so that it will retain for some time in the electronic energy states and it will come back to ground state, so that it will emit a radiation which will is called as a fluorescence. So this fluorescence spectra is clearly explained by Jablonski, hence he is known as a father of fluorescence spectroscopy, and this diagram is known as a Jablonski diagram. So in the Jablonski diagram, we can see electron is exciting to the excited states, and while coming back to the ground state, it will emit the radiation which we will call as a fluorescence. As we can see here, the transition is happening from S0 to S2 or S0 to S1, and this is for absorption spectrum. And whenever there is a transition from S1 to S0, then it will be called as a fluorescence. And the respected characteristics times are also there on the screen. So this is the fluorescence. So here what we can understand is there is a shift between the absorption spectra and emission spectra. This is because whenever the electron is excited to the higher electronic energy states, its energy is lost in the vibration energy levels. So the emitted wave or the fluorescence light will have a longer wavelength than the incident light. So the shift between the absorption spectra and emission spectra is called as Stokes shift. So this is a confocal microscope, and the concept of confocal microscope was originally developed by Marvin Minsky in and around 1950. The confocal actually defined as having the same focus, because in the confocal microscope the one focal plane of the sample is focused and the light will be collected from the same focal plane, hence it is known as confocal. So for this, this instrument uses special filtering techniques so that the unwanted light can be eliminated. So this is a layout of fluorescence confocal microscope. So here there will be a laser source, and which will be passed through the excitation filter, and later it will be incident on the dichroic mirror, and the light after which after falling on the dichroic mirror, it will be fall on the specimen, and the light will be collected from the particular plane of the sample, and so that the light from the other planes, which is the unwanted light, which will be avoided by pinhole detector here. So the signal will be, the required signal will be detected by the photo multiplier detector. This is the scanner head unit, and also we can see the optics behind this. So this microscope has excitation filter, Actually it is a band pass filter, so it will pass certain wavelength of light so that unwanted light or unwanted wavelength will not fall on the sample. So this has emission filter, which is a long pass filter, so that it will pass only a longer wavelength and it will block the shorter wavelength. Hence the incident light will be blocked and transmitted light from the sample only be allowed to pass through this filter. So this is one of the filter in the confocal microscope, that is neutral density filter. Actually this is in a neutral grain color, and this is actually designed to reduce the transmitted light intensity evenly across a portion or the entire wavelength spectrum, so without introducing any significant change in the color temperature. So in our instrument we have ND10 neutral density filter, and here if we have ND70, so the meaning is like if 100% of light is falling on the sample, then only 70% of light will fall on the sample and 30% of light will be blocked by this filter, so that the sample can be kept for very long time. Dichroic mirror is like a heart of the microscope. So this will produces the transition between the transmitted and reflected wavelengths. So it will allow to pass only the transmitted light. As you can see from this, so this is for excitation. So there is a band pass, it will act like a band pass filter. So it will pass only a band of wavelength and it will allow another band of wavelength here for the emission spectrum. So this is the galvano scanning mirror. Here we have lasers. So the laser has to scan the entire sample, and this is done by this galvano mirrors. So these galvano mirrors will rotate in x axis and this will rotate in a y axis, so that the laser will fall on the sample, fall on the entire sample, so that entire sample will be scanned. This confocal microscope is also equipped with the photomultiplier tube. Since this confocal microscope is using the laser light, it will falling only on the particular point on the sample, so that the signal coming out of the sample will be very less. So the signal has to be amplified and it is done by photomultiplier tube. So this photomultiplier tube will amplify the photon signals. This is again the heart of the microscope, pinhole aperture. So the light will be coming from the different planes, but this confocal instrument collect only the light from particular plane, and the stray light or the unwanted light is blocked by pinhole aperture, so that image obtained in the confocal microscope will have very high resolution. So this is the specification of FE3000. This microscope from Olympus, and this is the inverted microscope, and this microscope has 6 objectives: 10x, 20, 40, 60 and 100x, along with 4x, and this uses 4 excitation lasers, and the maximum thickness of the sample should be 200 micrometer, and this microscope has resolution along xy is 250 nanometer and along z it has 500 nanometer, and this has equipped with the 2 scanners, galvano scanner and a resonance scanner. And in this instrument we can place the slides, because slides holder will be there, and microteter plates we uh, can accommodate by this instrument, and we can also place the petri dishes, and different image techniques are used here: confocal, time lapse, Z-stack, 3D movie, fret and fran. In this instrument we can keep samples like slides and petri dishes and microteter plates. So this is a holder to keep the slides. So these are the 2 holders to keep the petri dishes, and we can directly keep the microtreated plates in this holder. So this is the one of the standard sample. We will place a sample in the slide holder. Once the sample is placed in the sample holder, we will focus the sample, then we will do the imaging with the software. So now we are in ocular mode. So now we have to switch to LSM, that is laser scanning mode. So in the meantime, the laser light will be focusing on the sample. We need to set some parameters before we capturing the image. So here we have two scanners, Galvano and a resonance scanner. So Galvano will scan the sample very slowly, and if we kept the resonance scanner, it will scan the sample very fast. So if I kept Galvano scanner, the time taking for frame is 2.143 seconds. So if I switch to resonance scanner, this will take only 65 milliseconds. So right now I only work with the Galvano scanner. So these are the two modes. One is one way and round trip. So one way will take more time and round trip will take lesser time. So as you can see, when I switch to round trip, it will take only 250 milliseconds. So the interlace tells how fast we have to scan the sample, and that will work for only round trip. So as I switch to 4x, you can see the changes in time taking per frame. So I'll keep the scan size. There are different scan sizes you can select here. So right now I'll be keeping it in 1024 in 1024. Then I'll go to PMT, that is photo multiplier tube setting. So under the photo multiplier tube settings, we have two modes, VBF and lambda. So VBF refers to variable band filter. So which means that we will tag the sample with the fluorescent dye, and will excite with the particular wavelength, and we will collect the light so that we can get the image here. So in the lambda, if you switch to lambda mode, we will excite with one dye, one wavelength, and we can collect the light in the entire visible range. We can move this band in the entire region. So this is average, that is line and frame. So we usually keep it in a line or anything we can keep. So if we keep line, so the sample will be scanned along the line and it will be averaged so that it will form the image. And if we keep the frame, the sample will be scanned throughout this frame, then it will be averaged. So that if the signals are not properly coming from some areas, so on averaging we will get a better resolution. So this is a sequence. So this sequence has to be given when we have more than one dye and their excitation wavelengths are very nearby. This is the dye and detector settings. Here we need to select the dyes. So this indicates the excitation wavelengths. This is the emission wavelengths. So first of all, I will add a dye which is having excitation wavelength 488, which is suitable for my sample. So I'll add this. So this is a HV. This is the voltage given to the detectors, and we'll always keep the gain as a 1, and we will increase the offset. Once we focus the image, we need to set the offset. So offset will remove or nullify the background. We need to keep it in a high-low mode. So this is the best condition to acquire the image. So we'll stop it here. we acquire the image. So this is the acquired image and this is the fluorescent image. Now I select 2 dye at a time. I have chosen the 2 dyes. I'll go live. We can choose the pseudo color. We need to set the offset. Now it is ready to take the image. So these are the merged image. This is the merged image, and here sample is tagged with the 2 fluorochromes. So at a time we can capture the image. So this is a merged image. These are the individual images. This is to capture the fluorescent image. So with this we can do time lapse. For that we'll keep time on here. We will select the some cycles. Minimum is the 2 cycles. So I'll take 3 cycles. Image acquisition is completed. So you can see here we have captured 3 images and which is moving here. Since the images are stationary, you are not able to find the moving images here. We can also do Z. So for that we'll keep on Z. Then we go live. We will go to one extreme end until the sample is defocused. So we'll register that position and we'll again come back to another end of the sample until again sample has been defocused. We will register that position, then we optimize it. So it will take 40 slices. So then we stop live and we'll do LSM start so that it will acquire the image. Here you can see 40 slices are there. Now it is scanning first slice. So scanning is completed. So this is the 3D image of the sample. so that we can rotate, we can see it. Also we can see the information along this line on this side as well as here. So finally we have shown how to take the image of the fluorescent samples, and also we have shown time lapse and Z.